So Robert had the title of the History of GMO Commercialization, and I've added a subtitle here, Pre-Market Safety Studies that illustrate the imprecise aspects of genetic engineering. I like to call it GE instead of GM because GM, modified, makes it sound more like it's more related to traditional breeding than I think it is. It's a very different process, which I'm going to talk about tonight. All right, so as Robert said, I worked on bringing the first genetically engineered food to market. I was working for a small company called CalGene in Davis, California. I came in 1988. They had already started working on what was going to be the Flavor Saver tomato. I was working on a separate tomato project at the time, but when the Flavor Saver, it became clear to us that we were going to beat Monsanto to market. We were going to have the first product. We scientists got pulled off of our projects and were asked to do regulatory safety studies. And so that's primarily what I'm going to talk about tonight. Now, you might say to yourself, this is a 20-year-old tomato. It was on the market for a couple of years. It's no longer on the market. It hasn't been on the market for a long time. The genetically engineered trait didn't really work very well. That was one reason the company went under. Monsanto bought the company a couple of years after the tomato went on the market. This all in spite of the fact that it was very well received. The local grocery store in Davis, California couldn't keep them on the shelves. In fact, they rationed them. You could only purchase 2 Flavor Saver tomatoes per person per day for a while there. And during the holiday season, the year they came out, they made gift boxes of them. So we didn't have, and they were labeled, as you can see here. And I'll show you a close-up of a sticker later in the talk. So why do I do this? Why do I talk about this? Well, the reason is because we were first, because we went to the, I'm going to talk mainly about what we did with the FDA. We set the precedent. Many of the techniques we used and the issues we had to deal with in trying to convince the FDA that our tomato was safe are still the boilerplates that many of the companies out there still use today. It's amazing to me how relevant this tomato still is to the GMO debate. So that's one of the reasons that I talk about this. Another reason I give talks like this is because most of the proponents of this technology that you'll hear give talks don't mention, they neglect to mention these issues that that I'm going to tell you all about today. Issues of risk and of concern, and that's not kosher. We need to tell all about the technology. The only way societies can deal with a new technology is if they have all the information and we can look at where the risks are, where the benefits are, and figure out how they should be regulated, and maybe even consider whether we should use the technology at all. Just because we have the technology, maybe we're not ready to use it. And that's actually what happened to me at CalGene when I was there. I was one of those folks who read Francis Moore Lepay's Diet for a Small Planet when I was in high school, trying to decide what I wanted to do with my life. And feed the world, I figured photosynthesis. I'm going to be a scientist. I'm going to study photosynthesis. That way I could come up with an alternate fuel, or I could come up with a food that could feed the hungry. And a lot of my colleagues who are very big proponents of this technology felt the same way I did and went into this field for the same reasons. But I, in this process of going through and doing the safety studies for this tomato for the FDA, I realized that maybe we didn't know as much as we needed to know to bring this technology to market. And I'll be curious to know how you in the audience feel about that when we're done here. All right. So this is one of my scientific heroes, Richard Feynman. I found that when I started questioning my going into genetic engineering as a result of doing these FDA studies, I started getting more interested in how we bring new technologies out of the lab and into the marketplace. And this man, who was awarded a Nobel Prize in physics for his work in electrodynamics or electromagnetism, I think it was, but also worked on the Manhattan Project and developing the atom bomb. So he thought a lot about this. What we do in the lab as scientists versus what happens when we take that science out of the lab and out into the marketplace as a technology. And he defined the two. There's basic science that you do in the lab to find out things, and then there's technology, which is using that science to go out and try to solve problems in the real world. And that, he said, is something, regulating technology is something scientists don't know that much about, and maybe scientists aren't the ones who should be telling societies how to regulate their technologies. But on the other hand, what he said was the role of scientists in this process of bringing science out of the lab is this quote, that scientists need to make clear all the information that is required for somebody else who's intelligent to make up their mind about it. And I feel very strongly that that's what our job is, the role of scientists. And I feel very strongly that a lot of scientists out there who promote this technology are not telling everybody all of the issues. And you can't just cherry pick when it's something as important as this. You have to, and that's not a very scientific thing to do, to cherry pick your data and just talk about the precision of a technology and not reveal all you know about it, things that society should be concerned about. So he's one of my heroes, and that's one of the reasons I'm giving a talk like this. All right, so like I said, at Calgene, there came a day when we were all called into a conference room and told to put aside our projects we were working on and that we're going to conduct these studies to prove that our tomato was safe and to be able to prove to the FDA that our tomato was safe. And so we established a dialogue with the FDA scientists, and it went on for 4 years or longer. And what we were doing was educating those intelligent people about this technology so they could make decisions about their policy and how they were going to deal with these products once they started coming out of the lab. So one of the first things we talked to scientists at FDA about was the difference between traditional breeding and genetic engineering. And I put some of those differences here on this slide. So traditional breeding has evolved over eons, and it occurs only between closely related organisms. Genetic exchange occurs in reproductive cells specifically, and it occurs between related chromosomes in those cells, and through homologous recombination, and homologous meaning related sequences in the DNA. And the amount of DNA, so as I'll show on this next slide, here's a little cartoon of what traditional breeding is all about. So what ends up happening is you start with the same amount of DNA. You end up with the same amount of DNA in a chromosome as you started with. And you exchange DNA among like chromosomes. So this sequence that has been exchanged between the chromosomes here encodes essentially the same genes as the segment that it's been exchanged with. Okay, so there's something we call it synteny, that the genes are aligned in the chromosome, in this chromosome, in the same order as they're aligned in that chromosome. So and this process, as I say, it's evolved over eons, and so along with the process are also pathways that ensure that this process takes place correctly. And if it doesn't take place correctly, those chromosomes can get kicked out. That cell may die, and it won't go on to the next generation. Okay, now this is as opposed to genetic engineering, which is a man-made process. It takes advantage of, well, some of the processes take advantage of things found in nature, but it's manipulated by humans without this evolution and pathways that ensure that everything happens correctly. It can happen, you can use any gene. You don't have to come from an organism that's related to you. You can, many of the products out there are bacterial genes being put into plants. And, you know, the being doesn't have to be alive. I knew a graduate student when I was in graduate school who wanted to get DNA out of a stuffed, extinct animal. And I think it was a quagga, which is like, looks like a zebra or, you know, part zebra, part horse. And so he practiced on leather shoes. He would grind up leather shoes to get the DNA out of the leather. You know, so you can take a gene from anywhere, or you can make up a gene in the lab, right? So all bets are off. You don't have to be related to anything. It occurs in somatic cells. We would take tissues, plant leaves, different parts of the plant, and insert those genes in those cells. The insertion for both of the processes that have been used over the last 20 years, the Agrobacterium tumefaciens method, and that's based on this bacterium, a soil bacterium that's found in nature, and it actually inserts, it causes crown galls in certain plants by inserting some of its own genes into the plant's DNA and getting the plant to make its food, basically. But there's no direction to that. It gets inserted here, there, everywhere. Not that nice homologous recombination that occurs in traditional breeding. Insertion, so it's randomly, and maybe because it's in somatic cells, we're not sure. Your gene can then randomly land in one of the plant's genes and mutate it. So if you land in a gene, you disrupt the gene, that gene no longer functions, and you've got a mutation on your hands. And that insertional mutation occurs at a very high rate. I think that's a very high rate. 27 to 63% of the time, you land in a gene in your tomato or whatever you're trying to transform. Plant scientists use the Agrobacterium method to try to, on purpose, mutate every gene in the model plant Arabidopsis so that they could then go in and pull that gene out with a tag that they had put on this tDNA, the transferred DNA. So they could try to make a mutant in every gene in the plant to see what those, as a way of figuring out what the function of those genes are. So that's how efficient this mutation process is. All right, and then the gene spacing. If you can imagine, if you're putting in a chunk of DNA, the genes that used to be next to one another are no longer adjacent to each other. And there are some examples in other higher organisms where altering the spacing between genes can alter the expression of the endogenous genes. You can upset what's going on in the plant without regard to your inserted gene. Also, I just add here that most times it's a very inefficient process to get a gene into a plant correctly and have that plant be fertile and able to be traditionally bred with other plants. And so we use, because it's so inefficient, we use these selectable marker genes. When we were doing this, and these genes are still being used, they encode resistance to an antibiotic. And I have to tell you that the AMA, the American Medical Association, is not happy about that. They don't think it's a good idea to use antibiotic resistance genes, and the World Health Organization has made statements, similar statements. this isn't a good idea, and they say, we'd like you to see this technology evolve so that you're not using antibiotic resistance genes any longer. But I know that genetic engineers continue to do so today. The vitamin A banana, for example, has a kanamycin resistance gene in it. All right, so here's my little cartoon about, so you end up with this is not to scale, but you do add DNA to the organism. It's got, this chromosome has X amount of DNA, and this has X plus Y. All right, so it's not the same. Now, really, genetic engineering should be thought of as several processes. So there's one process through which you take those somatic cells and you insert genes into those cells. All right? But then you've got a plate of cells with a new gene in them, and that is not a product. That's a petri dish, and you have to take those cells that have the gene inserted into them and go through step 2, which is plant regeneration. You have to coax those cells into developing into a whole, fertile, normal plant. And that process is a stressful process, as you might imagine, going from one cell to a whole plant. And there's a mutation process that is referred to as somoclonal variation, where you get changes in the DNA, and you didn't do it on purpose. You don't know where they are, or we don't know a whole lot about somoclonal variants, but it is a kind of a mutation process. And then just this stressful process can also induce mutations of its own. It can set, for example, transposons into action. You can have silent, these little, well, Barbara McClintock worked here at Cornell for a while, and she discovered these jumping genes, they called them. And they can be quiescent in a genome, but under a stressful situation, like regeneration, they can be induced to be active, and they go around jumping into genes and causing mutations. And then after all that is done, you have a nice-looking plant, and it's got your gene inserted into it. You still have to undergo the traditional breeding process. So, and you don't have a product when you get that regenerated plant. That has to be traditionally bred. Usually the varieties of tomatoes and other plants that can be regenerated go through this regeneration process. They're not the kind of crop you want out in the field. So you still have to do traditional breeding. All right. Now, specifically to Calgene, what we were talking to the FDA about. Here's a cartoon of the vector that we used, and a vector plasmid. That's just really a name for a circular piece of DNA. And this one is the one that was in Flavor Saver tomatoes. So what we've got here is, this is the agrobacterium method. And again, as I said, the way that works is that this bacterium has this plasmid that it inserts its own DNA into the plant. So what we genetic engineers do when we use this method is, as long as we have these border sequences here, we get those from the agrobacterium, then we take out the genes from the agrobacterium and insert in place of them our genes of interest. And then we just ask the bacterium to go through its normal process of inserting that DNA, and whatever's between those borders is what's supposed to be put in, it is what gets put in the plant. And there had been 10 years of plant molecular biology research that had occurred by the time we were doing this experiment, and everything said only the stuff between the borders gets inserted. So that's where you put your genes of interest, and in this case we have our antibiotic selection marker gene over here, the kanamycin resistance gene. And then we've got this, we used the cauliflower mosaic virus, this promoter, a region of a gene that turns the gene on and tells it where to be turned on, when to be turned on. And most genetic engineers use the cauliflower mosaic virus 35S promoter because it's on at a high level everywhere. And if you just want your gene to be screaming on all the time, that's what you use. And that's what most products have this particular promoter in them. So we're using it to express, to turn on our flavor saver gene. And what that is, it's fairly relatively innocuous, or we used to think so at Calgene. What we did was we took a gene out of tomato that encodes an enzyme that softens the fruit, that is involved in softening the fruit. And we flipped it upside down and backwards and put it behind this cauliflower mosaic virus 35S promoter. And the idea was that this was an antisense strategy, that somehow this message, this antisense message, would find the regular polygalactoronase message out in the cell and glom onto it and prevent that message from being made into the protein, the enzyme that would then soften the cells. So it was a way of shutting down the expression of the tomato gene. Does that make sense? Okay, so that's what we were trying to do. And then, so what we did was we very carefully for the FDA characterized all that DNA that was between the borders. We knew every DNA sequence there. We had put it together. We looked for any reading frames, any other thing that could be encoded in this DNA. We ensured the FDA that only our selectable marker gene and only our flavor saver gene would be expressed if we put this sequence of DNA into a plant. All right, and that's important to remember because I'm going to tell you about a surprise we got about that in a bit. All right, so my job with the flavor saver tomato was, all right, we want to not only educate the FDA about how this technology works, but we want to demonstrate how precise it is. If you hear proponents talk about this technology, precision is a big aspect of it. This is so precise. All right, so my job, I was given the job of identifying plants that had one copy and only one copy of the DNA we meant to put in the tomato. And my boss said, I'd really like you to have 10 plants, 10 individually transformed experimental plants that one of them might go to market, and that's what we want. So I spent a lot of time. Oh, and the numbers on this. So I was telling you about it's an inefficient process. At Calgene, we started with 21,250 experiments. Little pieces of cells, callus tissue or leaf tissue that had been transformed. And by the time I was, and then you see how many genes have been inserted, how many are make seeds. You know, some of them don't, after they come through tissue culture, they're not looking too good. You throw those out, they're never going to make it to market. By the time it whittled down to, oh, and you have to shut off that PG enzyme, right? You have to be very efficient at shutting down the PG enzyme. So I had 44 plants out of the 21,000 that by the time I started my project. And at the end of looking, some of them, it turned out, actually had the gene inserted in multiple places in the genome, and we threw those out. We wanted them inserted in only one place because, for example, that limits the amount of insertional mutation that you can have, right? If it's only in one place, you only have a chance of inserting into one gene. So we threw out everything else, but when all was said and done, I had these 8 plants. These represent 8 plants, and not a single one of them had only one copy. Every one of them had either 2. So this is one copy, and this is another copy. We had this one that had 3 copies. And then this one was the closest we came to having only one copy, and it still had a smidgen of a copy on this side. So that was a surprise, you know. But my boss said, okay, this is what we got to work with. This is what we're running with. And it turned out that it was not this process that we thought, this antisense process that I described earlier, with make the whole message and glom on to the antisense version of it here. it was another process completely. And maybe if we had worked a little, and we had received a patent for this anti-sense process, and well, the patent office believed that this was the way it was going to work, and we thought so too, but that's not the way it turned out. Years later, this paper was published in, so we're talking originally the early 90s, I'm doing these studies. And this paper came out in 2008, and they went back and looked at our tomato, and they decided that no, this was another technology called RNAi interference. And several years after our tomato went on the market, Andrew Fire discovered this RNA interference technology, and he eventually got the Nobel Prize for discovering this and looking into the mechanism of it. And so what we think happened is we were inadvertently setting it up with RNA interference. You put in the same construct 2 copies, and this will get transcribed into a message, and it flips over on itself. And you have, instead of trying to find something out in a cell milieu, you have your double-stranded RNA, which can then go and shut off the gene that corresponds to the sequence you have the double-stranded RNA for. I'm not an expert in RNAi, obviously, but if we had looked a little harder, we might have gotten a Nobel Prize for this. But basically, we didn't know what we were doing, right? We didn't know the mechanism that we were shutting down this gene using. All right, and Bill Hyatt and Rick Sanders were my colleagues. They worked on the original tomato project with me. They stayed, after Monsanto bought our little company, they stayed along for the ride and worked at Monsanto for a while. Okay, so that was a surprise. We couldn't find a really precise example of our tomato gene. And then another surprise was, and this one was the one that really got me reevaluating what we were doing and my feelings about the technology. As I said, I was gung-ho. This has so much promise. This is so powerful, this technology. But the FDA at one point came to us and they said, you know, you tell us that only the DNA between those tDNA borders is going to be inserted into the plant. You know, how do you know that? And I was with my boss at the time, and we both said, oh my gosh, you know, 10 years of the scientific literature says that's the only thing that goes in. And so I did basically a literature search, and I went to an expert in agrobacterium, Patricia Zabriskie, a professor at Berkeley, and she had a brand new review article out that described it. Only the sequences between those borders are going to be inserted into the plant. And so that's what I sent back to the FDA. The FDA came back to me a month later and they said, no, we didn't ask you for a literature review. We want you to look in your tomato plants and see if any of the other vector sequences are in there. So Rick Sanders and I, reluctantly, we thought, oh, that's going to be a messy experiment. But we carried out the experiment. And lo and behold, 20 to 30% of the time, the whole rest of that vector that we didn't characterize for the FDA had gone into our plants. And there was an additional antibiotic resistance gene out there and just a lot of uncharacterized bacterial DNA. We had not intended for any of that to get into our plants, and there it was. And all we could do was say to the FDA, well, you know, this happens. And so what we're going to do is we're going to look for it every time we make a genetically engineered plant, and we're just going to throw out the ones that have this inserted into it. Now, recently, I took a tour of a small biotech company, and I ran into a former colleague of mine, and I was told that when she arrived at the company, they were using the agrobacterium method, and they weren't looking for extra DNA sequences. And she said, oh, no, you got to do that. So here's my paper. When we found this out, I I said to my boss, we have to publish this. You know, this isn't just something we tell the FDA, because this is going to affect the work of scientists in the lab, academic scientists. And he didn't want to do it. You know, he's like, we're going to keep it a trade secret, or I don't know what, you know. But luckily, our CEO and the upper management said, no, we're being transparent about this process. We're going to FDA when we're not even sure we have to, and we're going to publish. And so, but because my boss, Bill Hyatt, was so against the idea, I wasn't allowed to use the word tomato in my paper. So if you look up this paper, you won't find the word tomato in it. But we also, it wasn't just our vectors. I tested vectors we had gotten from other companies. It wasn't just tomatoes. In our rapeseed plants and our cotton plants, we found this to happen also. This is a problem that's still out there. Nobody has found a way to get around this. It happens, and this is one of the reasons I think that regulation of these products at the FDA should be mandatory. Right now it's a voluntary process. You make a genetically engineered plant, you don't even have to go to the FDA if you don't feel like it, in most cases. And this is one of the reasons, this and insertional mutation are one of the reasons that it should be mandatory. On a case-by-case basis, they should all be regulated. All right, so that was 2 surprises. Then, because of this insertional mutation issue, where you might do something to a plant that you don't intend to do, if your insert if your DNA genes end up in another gene in the plant. So the FDA came to us and they said, you know, we think you should do some animal feeding studies. That's a way that you could look to see, make sure if the animals are okay, if you, after you make them eat flavor saver tomatoes all day, or for 2-3 weeks, or 6 weeks, or however long, that'll be reassuring that there were no unintended things happened. So we did a few experiments, and the first ones looked great. You know, we get 0 out of 0. We're looking for gastric erosions. Because, all right, I'll give you a little background on this. When I left for maternity leave, we were going to market with a couple of different events. And when I came back, they had chosen 2 new events. And so they set up these rat feeding studies with the 2 events. I came back from maternity leave. My boss takes me down to the lab. He says, you know, here's a pile of tomatoes. I want you to wash them really well, and we're going to send them out to the company that did the rat feeding studies, and we're going to do the studies again. So I said to him, why? You know, why are we going to do them again? And he says, well, there's been a problem with the rat feeding studies. And as you can see on this slide, okay, so in this slide, this is basically a screenshot of the FDA website where you can go and read about the Flavor Saver tomato. There's a food master file containing this information on the FDA's website. So it was one event, this guy. So CR3, 623, in only the female rats, right? 4 out of 20 female rats had these little erosions, like lesions in their stomach linings. And so, you know, we're all upset about this, like, oh my gosh, what's happening here? And so we called in another group of pathologists to blindly go through the slides again and look at the slides and the tissues from these rats that had been fed this particular, well, all of them, actually. They looked at all of them. And instead of, you know, not finding the lesions, which was what our wishful thinking was, they found more. So the second time, a group of pathologists found 7 individual female rats who had these lesions in their gastric tissues. So we repeated the test. You know, okay, that's not good news. What do? All right, we're scientists, so we're going to repeat this experiment. And that's what my boss was asking me to do. You know, we're going to send all these tomatoes, this variety and some other controls, and we're going to feed them twice as much Flavor Saver tomato. We're going to see if there's a dosage effect. Do we get more of these lesions if we feed them more tomato? That would indicate, yeah, that probably is as a result of that tomato, right? So that's the proper scientific response. And one reason I bring that up is because there have been some surprises out there in the scientific literature. Eric Seralini's paper, for example, where they're looking at the rats fed the BT corn, glyphosate-tolerant corn. Are there any unintended things happening if you feed it to rats for a while? And Seralini found that looks like maybe there could be. So the proper scientific response to that is do the experiment again, right? It's not call up the journal and have them retract the paper, which is what happened, right? All the proponents are up in arms. Oh my God, you know, isn't it terrible? When the editor of the journal went through the data with a fine-tooth comb, and the worst thing he could say about it, this is after it had passed the peer review process, it had been in the scientific literature for over a year when it was finally retracted by the journal, and the worst thing he could say about it was, you could use a few more controls, you know? And so I actually emailed the authors of papers at Monsanto and said, so you guys are going to do that, right? You're going to repeat the experiment, right? He's looked through all the data, just do the same thing, use more control rats, maybe use that other variety of rats that doesn't get the tumors as much, or use both, right? Do a big experiment, put this thing to rest. Oh no, we're not going to do that. The Monsanto guys tell me, we're not going to do that. The FDA doesn't make us do that. Why should we do that? I mean, this is a case where I really think the FDA needs to come out and say, we told you that it's your responsibility to make sure your products are safe. Looks to us like you need to do some more experiments here. Anyway, we did more experiments, and I have to say, we didn't really get to the bottom of this erosion business. And the FDA just stepped back and said, you know, we're not going to require anybody to do rat feeding studies. So, and it looks like these happen in the water controls once in a while. You get a few up here in the water controls. You get a few in the control tomatoes that haven't been genetically engineered. So we're going to say this is kind of an incidental thing. It happens sometimes, and we're going to let you go to market with this guy. So we went to market. We had the world's first genetically engineered whole food. We labeled it, as you can see. We were not required to do so. I've already told you how it flew off the shelves. It was popular. Oh, and by the way, in the transparency business, you know, not only did we repeat the experiments, the rat story got published in the newspaper. You know, we were completely transparent. We talked about everything we found. And the public didn't seem to have a problem with it, right? We were labeling it. You can make up your own mind. Buy it or not buy it. Be an early adapter or not. All right, and here's a close-up of it. This was a sticker that was on a package of cellophane-wrapped Flavor Saver tomatoes. And I just wanted to point out that right on the sticker, grown from genetically, and we use the word modified. I started using engineered later. But right there on the sticker, and like I say, the public didn't have a problem with it. Okay, so what's happened since the Flavor Saver tomato? I didn't talk about our selectable marker gene. We also did a bunch of studies, and I did a lot of those, on our kanamycin resistance gene, and submitted a package of data to the FDA. And we actually submitted a data package using an advisory opinion procedure that the FDA has. And basically the advisory opinion procedure is when something is applicable, widely applicable. And so this kanamycin resistance gene, as I said, is used by many companies, many different companies, and it's still being used by many developers of genetically engineered crops. And so that seemed like the right approach. But the FDA came back to us after we submitted our data package, and they said, you know, actually, some people deny that. They say, oh, Calgene wanted to do this. No, Calgene was asked to do this by the FDA. I sat in on the conference call when they asked, And they said, you know, we think that Congress will understand this process better, this whole area, if we use the food additive laws on the books. And originally, back in 1986, the Office of the President, when they first started talking about how we might regulate these crops when they start coming out of the lab, they said, we're going to use the laws on the books. We're going to use the laws on the books, and we're going to use the agencies that are already in place. We're not going to come up with some new system. We're just going to work what we've got. And so they decided, okay, a food additive, that makes sense. You know, this gene encodes a protein that's going to be in every cell in a tomato. That protein was never in the tomato before. Sounds like a food additive. I mean, that's sounded logical. We scientists thought from the get-go we should use the food additive petition process. But the FDA said, yeah, why don't you do that? So after my boss, our CEO, said, well, what's that going to do to my stock price? The FDA guy calmed him down and said, you know, you don't have to start your clock over. It's a similar format. Just put your data in this new different format, and you won't have lost any time. We'll review everything. And so we did. And they came out and they modified the food additive statute to declare the protein product from this gene a food additive in tomatoes, rapeseed, and cotton. Sometimes I wonder, you know, it's being used in a lot of other crops, and you know, shouldn't they have gone through the food additive process also? But they haven't. So here is another screenshot from the FDA's site. And if you go to read about biotechnology products at the FDA site, you will see that our tomato and this particular protein product is the only one genetically engineered product that has been treated as a food additive. After our tomato, nobody else did. And most of the products out there have a protein in the plant that has not been there before, or a lot of them do. But nope, they decided, nah, we're not going to use that food additive. Well, their system is voluntary. It's a voluntary process. You theoretically don't even have to go to them. Okay, so yes, we're rounding up here what has happened since the Flavor Saver tomato. I've already talked about the food additive part of it, voluntary consultation. And of course, the developers are the ones that provide the data for the most part and declare their products safe. And the FDA, they just receive a letter from the FDA saying, well, we have no more questions for you, and it's your responsibility to make sure your product's safe, and good luck to you. All right. Oh, this is to remind me to tell you that I've already told you that the Flavor Saver was a commercial flop, that the fruit didn't really stay firmer on the vine enough to let them ripen on the vine. The idea was to let them ripen on the vine themselves, and then they'd have a better tomato flavor like you get when you grow them in your backyard, but stay firm enough that you could still throw them in a truck and get them to market. And that didn't work out. We ended up buying all kinds of equipment for dealing with peaches, actually, to treat them with kit gloves. But now there are, so the one trait that they did have was this longer shelf life. So you could pick a ripe tomato and put it on the shelf, and 4 weeks, 5 weeks, 6 weeks later, it still looked great. And we have, Calgene used to have, they had a centerfold actually in one of their annual reports with the Flavor Saver tomatoes looking great and the pile of goo tomatoes next to them that were non-transformed. But this slide is to remind me to tell you that a longer shelf life tomato has been made through traditional breeding. And so you can do a lot of these, our tomato, so there's one out there, there's several. I think Israeli breeders came up with a mutation actually in the polygalactorejonase gene or another tomato softening gene that keeps them longer on the shelf. All right. Okay, and I've talked mainly about the Flavor Saver tomato, but I've kept up with the goings-on in the industry since I left Calgene back in 1995. And I was an author on this paper where they were using that RNAi technology I talked about earlier, RNA interference, to shut down a gene. In this case, it's Arabidopsis. And just looking at the construction of the vector. If we place our gene sequence closer to the promoter or further away from the promoter, does it work better or worse? And just kind of characterizing the process itself. And, but along the way, we found that this particular construct, if you use that cauliflower mosaic virus promoter to crank out those small RNAs, those double-stranded RNAs, you didn't only shut down the gene you were aiming to shut down, you altered the levels of endogenous small RNAs. So these small RNAs, it turns out, like this Nobel Prize I told you about earlier, there are systems in all organisms, actually, that shut down, alter the expression of genes. And it's through these small double-stranded RNAs. And in this case, we change the levels of the endogenous small RNAs. So there's this system in place to regulate gene expression, and we inadvertently changed the numbers of those molecules in the cell. So at the end of our abstract here, we say, in addition, if you strong, inverted repeat sequences affected the prevalence of these endogenous small RNAs, which has implications for the use of this technology in experimental or applied purposes. So we still need to look out for these things. You know, it's not a perfect technology. There are lots of these nuances. I've pointed out, several of them tonight. We need to have regulation of these things because it's on a case-by-case basis. And the World Health Organization, UN organizations say the same thing. So, all right, before I do that, I have to tell you that, so we need to look at them on a case-by-case basis. So anybody who says all these things are safe, I'm not even convinced that all of them out there right now because we still have that Seralini corn is still out there, right? That hasn't been resolved, the rat feeding issue. We've had commercialized products like Starlink corn, where it failed every test the EPA gave that the protein inserted into that corn was behaving like a human allergen. And the EPA said, well, we're not comfortable letting you commercialize this. But as long as you don't feed it to humans, well, we'll let you just commercialize it if you only feed it to animals. And so they restricted to feeding it to animals, and within a few months, an NGO went to the grocery store and looked at the taco shells, and sure enough, corn being wind pollinated, it's very hard to keep everything separate. There it was in the taco shells and other corn products. So that had been commercialized. And I'll tell you, the CDC and the FDA and the EPA were concerned enough about Starlink corn that they monitored the US corn crop for 7 years after the thing was pulled from the market to make sure that the levels were low enough that they could stop worrying about it. So maybe you could say everything out there, nothing's been proven unsafe, but the government definitely had a concern over that product. There's also been other products. There was here at Cornell, there was the paper in Nature about how BT could kill monarch butterfly larvae in the lab, right? And a big uproar again. Oh my gosh, you people are so negative about genetic engineering. And the response at that time, as opposed to the Seralini time, for example, was a good response. It was a scientific response. They got stakeholders together from the NGOs, from the academic side of things, from Monsanto and the big companies, and they all sat around and they said, how are we going to ensure that this isn't hurting monarch butterflies? And they designed a bunch of experiments. They went out in the field, conducted these experiments, published 4 or 5 papers in PNAS, but most proponents of this technology will tell you, and it was all safe. All those papers indicated everything was safe. Well, there was one particular product out there people were less safe. It was less safe for monarch butterfly larvae. BT-176. It cranked out such high levels of the BT protein that it's 100 times more toxic than any other product that was out there. So there are examples of commercialized products that have been less than safe. And so we still need to look at each one, All right. So I'm going to end with another quote from a scientist that you might have heard of, Albert Einstein. The right to search for truth implies also a duty. One must not conceal any of what one has recognized to be the truth. So there are truths about genetic engineering that are not being told. And granted, there are a lot of nuances. This is complicated technology, and every product is different. And it's hard in this modern world where everything's reduced to an elevator speech or a sound bite to get across all these nuances. But the devil is in the detail, and that's what science is about, the details. And it's not science-based to gloss over these details. It's not science-based to make generalizations about the safety of this technology. It's science-based to look at the positive things and the negative things. You have to not cherry pick what you tell the public. We've got to tell them everything so that we can come up with a reasonable way to regulate these products if we're going to use them. And I'll stop there and take any questions anybody has.
So did anyone ever test the nutritional value of those long-life tomatoes, any kind, GE or non-GE, after sitting around on a shelf, before and after?
Okay, that's a really good question. I'm sorry I neglected to, I concentrated on our surprises and not the stuff that happened the way we might expect. No, we definitely did. And the FDA made us do that. So if you're going to sell a tomato that's been sitting around for 6 weeks, we want to know that it's got as much vitamin A. Turns out that Americans get a lot of their vitamin A from tomatoes, so we eat a lot of tomatoes through processed products or other products. So yes, we did that. We did other studies. We also, after we put it in the first data package, we also made sure that once we got a gene into the tomato, that it behaved like any other Mendelian gene. So we looked at generation after generation after generation, and that gene stayed the same. It was passed down to other generations in the same manner. We also, the FDA made us look at any possible toxins in the tomato. Could the levels of those be changed? Because that's the idea that, say there's a gene, tomatoes make glycoalkaloids. Okay, glycoalkaloids are poisons, actually, depending on the particular glycoalkaloid or the levels of them. For example, that's why you don't eat green potatoes or potatoes with lots of sprouts. They tend to have these glycoalkaloids that have killed people. You can't cooking doesn't kill the molecules. So you have to be careful of that. So our hypothetical scenario to the FDA was, all right, we could cause these insertional mutations. What if there was a gene that green tomatoes make lots of glycoalkaloids, and then as tomatoes ripen, the levels go down? What if the gene responsible for the levels going down gets mutated? And then you would have high levels of glycoalkaloids in a tomato fruit when you normally wouldn't have that. And so the FDA made us look at glycoalkaloid levels. In fact, with our rat feeding studies with the lesions, we ended up doing so many studies with glycoalkaloids. And the trouble was not many people had been looking at glycoalkaloids in tomatoes. They were focusing on potatoes. that the methods had to be developed and had to be tested, and I spent a lot of my time learning about glycoalkaloids back in the day. But yes, they made us look at the normal vitamins, vitamin C, vitamin A, and the stuff you would expect, and make sure that they remain the same. But that's the trouble with the unintended, you know, it's limited by our imaginations. You know, what could we be screwing up in there? That's why they do the rat feeding studies. And as an aside on the rat feeding studies, you know, that's one thing that I followed the Seralini incident pretty closely. And most people, nobody really said we shouldn't be doing rat feeding studies. And the folks who are experts in rat feeding studies, like the editor of that journal, didn't say this was a stupid experiment, we shouldn't do that. So it seems as though, you know, these are valid experiments, and I think they should be required. If they're valid experiments, and we have unintended things we could be doing with this technology, we should be looking for, trying to identify something that might have happened.
I have two questions actually, Belinda. So the first one is, you said that at one point you sort of made an aside that bacterial DNA was inserted at the insertion events that you analyzed. Mm-hmm. And I wonder if you know exactly where that DNA came from, or if you can tell us more about it. The second question is, do you know why Monsanto bought Calgene if its product flopped and you didn't have any others?
We had others, is the answer to that second one. We had a big program in altering oils. That was what the Brassica projects were primarily about, that you could change the composition of the oils, playing with the genes, and make them more healthier oils. And there are products, Monsanto did put a product out that was a healthier oil. So that was the answer to the second. And the first was, when we did the experiment to look for the extra sequences, we used the extra sequences as the probe. So we did the southern blot, if you know that technique, and so we were looking specifically for, it wasn't just any old bacterial sequences, it was those sequences that we hadn't characterized very well at the bottom, at the bottom of that slide on that, and we found them. We found them all over the place.
So this is a different angle on what you shared. I'm interested in the psychology of your experience, in the sense that you started out, you shared that you had idealism was motivating your work, and that as you went through this experience and you began to be concerned, you began to maybe start to conclude that what you started out doing, thinking it was something positive in the world, actually may be harmful or moving in the wrong direction. What was that like psychologically? And as you shared that inner experience with the people around you, what did you see, what came back at you from the group?
Oh dear, you're bringing back up some really unpleasant memories for me. It was a process. So I started off in graduate school studying photosynthesis, like I had thought would be valuable to the world, right? And then I went and did a postdoctoral fellowship at the University of Chicago, and I continued looking into photosynthesis. And then I got this I heard about this job at Calgene, and I was also looking at how the cells involved in photosynthesis develop, and how more efficient systems of photosynthesis develop as opposed to less efficient ones. So development I was interested in. And they had a job looking, this tomato solids project I was working on was looking at development of a tomato fruit and what genes are expressed and how you might alter that development. So right away when I took the job, and one of the reasons I took the job was because my parents lived nearby and I was marrying a seismologist, and the company's in Northern California, and that's where he can find a job, and there are extenuating circumstances, right? So, but actually when the dealing with the FDA came up and establishing, I mean, we really felt like we were establishing the boilerplates, and we did. There's the companies out there, when they go to the FDA, they're still looking at the similar things that we did back at Calgene. We set the precedent for how you do these kinds of safety studies. And that's what I said to myself. I mean, you rationalize is what you do. You say, oh, at least I'm not working on that project. It's about herbicides, and they're going to spread herbicides all over the place. I'm doing my development of tomato. And then you say, okay, this establishing how these things are going to be regulated, that's important. That's important when, I'll never forget when we had our party after the Flavor Saver came out, and my boss said, well, you know, people are now eating our experiment. You know, it's kind of scary. So it was a big thing we were doing, we thought, to help the FDA educate them and identify the possible risks and establish the process that this stuff was going to come from the lab and end up on people's dinner plates. And I thought, even if I'm not going to feed the world, this will open the gates to other products that'll come afterward that could, right? And actually, and at Calgene, like I say, when we came up against a problem, we answered it with science. And I was happy about that. So I could raise my hand and say, no, I don't think that's the way to interpret that experiment, or we have to do this. And people would listen, and you know, you could speak your mind and give your ideas about science, and that was great. What wasn't so great was when we started interacting more with the business staff, and that's when I got really unhappy at the company. And I mean, we had a business guy come in, for example, and say, you know, could you scientists remove the error bars from your data? It's really confusing to me. And I'm like, you know, the error bars indicate that our product isn't any different than their product, and if we remove the error bars, it looks like our product is better, and that's not the truth. For seeking the truth, you have to have the error bars. So I was getting not as happy about interacting with the business staff, and there were a couple of times when I tried to quit a couple of times, and our CEOs told me, oh, you can pick any project in the company you want. And I picked a cotton fiber project that was as far from commercialization as any project in the company. So it was a process, but I was open, and I think being a scientist, you have to be open to the new evidence as it presents itself. And when your rats get lesions, or your vector goes in and you didn't expect it to, or you know, all of these surprises, you have to reevaluate. You have new information, you got to reevaluate where you stand. And that's what disappoints me about my colleagues, my former colleagues, or people who are so gung-ho about the technology, that you have to reevaluate it. You have to, if you're seeking the truth, you have to speak the truth about it. So I feel that it's taken courage to do this. I mean, I just had a Freedom of Information Act request last week. So somebody's trying to dig into my emails and figure out why I'm, you know, whatever they're trying to do. It's not, I had somebody tell me, you know, it takes a lot of courage to get up when so many people out there are just telling you about how great this technology is. But that's not the whole truth. We got to tell the abject truth. The truth, the whole truth, and nothing but the truth, not, you know. So I feel like I've stuck to my guns, and not everybody around me has. I think they're still so wanting to feed the world. I think they have all the best of intentions. They really, but you got to reevaluate and proceed more cautiously as a result. And I've been able to do that, and some people don't seem to be able to.
Maybe I missed it, but I didn't understand the real reason that the tomato didn't succeed in the market. Was it because the shelf life wasn't as good, or because people started hearing some of your results?
I didn't explain it very well. Well, the real trait, it turned out, was the shelf life. But that wasn't sexy enough for the business staff. They wanted to have a fresh market tomato business. And shelf life is not that. They wanted to be able to let the tomatoes ripen on the vine. So if you let a tomato just get a little bit of color on it, a little bit of orange or red, and you pick that, it'll ripen on its own. I've even picked some green ones and they'll ripen, but they have to be kind of ready to be showing some color other than green. And so that was where they felt they did their business, analysis, and that was where the big money was going to be. If we had a more flavorful tomato, because we could let it ripen on the vine and it'd stay firm enough. So basically you have these two stages of tomato firmness. You have it while it's ripening, and then you have while it's essentially rotting. And what the Flavor Saver ended up doing was preventing it from rotting. It didn't really make it firmer on this side. It was just we looked and looked and looked, and in a couple of tomato varieties, it seemed like maybe it was helping a little bit on that ripening side. But for the most part, we lost money. We sold the tomato for $2 a pound, and the rumor around Calgene at the time was it was costing us $10 a pound to get them to market. So we were losing money hand over fist, and we tried with the peach equipment and this and that. And basically, we had staked the whole company on that product. It had taken so long to get to market that we had a couple other products in the pipeline, but Monsanto wasn't interested in the tomato. They bought us hook, line, and sinker and booted the tomato and went with the oil projects. But that was it. It really didn't pan out, the trait. The trait did the one thing, but it wasn't what we wanted to make a business around.
But how did it taste?
Well, it was mixed. And it turns out tomato flavor is a very difficult thing to study or to maximize or anything. The best traditional breeding program in the country at the time was a fellow in Florida, and I've forgotten his name, but his system was, he had the best tasting tomato varieties out there, and this was his system. He liked tomatoes that tasted more sweet. And he had a partner who liked tomatoes that tasted different, less sweet. And so they'd go walk through the field and taste all the tomatoes, and if only one of them liked it, they threw it out of the program, and if they both liked it, they kept it in the program. Now, when I was at Calgene, that and the way we measured softness or firmness was by setting a tomato down, and we had this little rinky-dink setup where you put a lead weight on it, and you gave it a certain amount of time to see how much it would squish, per time unit. And my boss hated that. Here we're this high-tech company, and we're using this thing, and we can't even measure tomato flavor. And so they put lots of money into that too. How are we going to get some kind of quantifiable tomato flavor? And we were holding focus groups and doing all this stuff. And basically, we were a bunch of genetic engineers, gene jockeys, and we were trying to understand farming, and they were doing it from what do they call it when you start to finish? Instead of doing a little boutique, we'll give you this. They wanted to do the business from the ground up to the consumer, and we just didn't have the experience to do that, and we weren't large enough, and so Monsanto took us over.
I just want to insert a little anecdote. I was at UC Davis in the genetics department.
Oh.
And there was a tomato breeder, Charlie something, I can't remember.
Charlie Rick. Yeah.
And legend had it that the way they selected for tomatoes for harvesting on the mechanical harvesters so that they would make it to the cannery was the graduate students would throw them against a wall. And the ones that survived got bread.
I have another Charlie Rick anecdote. He went and collected tomatoes from their origin, which is in Peru, in the mountains of Peru, and South America, other places in South America. He'd go and collect them all, and they have a seed collection place at UC Davis. Well, he used to collect them in toilet paper rolls, for one thing. So he would set the seeds down and then roll them, and he'd have a roll of toilet paper that contained all these seeds. But he was having trouble getting some of them to germinate, and it turned out that one variety in particular would not ever germinate unless a Galapagos tortoise ate them and they came out the other side. That was when you could germinate those seeds. So, and when we were first doing taste tests, I mean, so it was difficult to do taste tests until we had regulatory approval, right? So you're trying to get this thing ready for market, but you don't know how, you're touting its flavor, and the FDA actually said, can you really say that it has, you have to show us that it has better flavor. I mean, you can't just claim it does, you have to, so, but before we had USDA approval, for example, we couldn't eat this, we couldn't eat a tomato because we would scatter the seeds, right? So we had to take the seeds out, and but if you don't have all the pericarp and all the parts of a tomato, you're not going to get the whole flavor components. So that was an issue.
So this is maybe a little aside, but what happened to all these people in the FDA and USDA that seemed like when you were working at Calgene, these people were actually asking you the right questions and willing to work with scientists? It seems like those people have gone out the revolving door and maybe didn't come back in.
Yeah,
or something.
that's a really good question. And, I think Stephen Drucker could answer your question better, and I've talked to him about this kind of thing. So I was only dealing with scientists at the FDA, and like I say, they were asking great questions. I mean, that question about whether the extra sequences would go in, I never would have thought to ask that. That was a really great thing. I thought they were on top of it. They were asking us good questions. They were following up on things. I think, and then they came out with their policy in May of 1992 before they even got back to us about all these data we sent them data packages like this, and they hadn't gone back to it or resolved it, right? It was still in progress when all of a sudden they say, oh, Calgene doesn't have to do that, and we're going to have this voluntary consultation process, and this is just an extension of traditional breeding and all this stuff. And I'm like, where did that come from? I mean, that was another thing I took a step back, and okay, we're doing this in good faith, and then this is the result, that they're not even looking at. So. But I think Stephen would say, he did a Freedom of Information Act with the FDA and got the memos that went back and forth to the scientists and the policy people at the top. And the scientists were saying, this is different from just traditional breeding. This, there are unintended things that could happen. And it was, to me, and I think that's Stephen Drucker's conclusion, is that the policymakers just said it was kind of a political decision, basically. And there were, in my book, I found a source at one point where somebody in the Clinton administration said, well, this is the spin we're putting on the technology. It's the spin. And I wrote in my book, I don't think the American people want spin about this technology. I don't think that's the way to go. So there was a disconnect. I think the scientists and the FDA were doing a good job, and then something was going on at the political policy level that negated all the science, basically. So when people say, oh, it's science-based, that we have a science-based, regulatory system, we don't. That's not science-based. Science-based would be taking into account these issues I've been talking about tonight. It's not science-based. I hope I've demonstrated that I love science. I am a scientist. I take those personality tests, and I'm like, science, 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 So another reason that I give these talks is not because Richard Feynman thinks I should, or, Albert Einstein thinks I should. But I'm afraid that not being as clear, not doing our job as scientists and explaining the technology, warts and all, is going to come back to bite science. That it's going to give science a bad name in the end. That we need to be completely transparent. And yes, it's difficult to explain the technology. The devil is in the details. But that's what science is about, and that's what we need to try to do. I'm going to a conference, a workshop in December in Washington, D.C., And we're going to talk, they're getting lawyers and journalists and scientists together about how to communicate scientific uncertainty to the public and in general. And it is difficult. It is difficult. But we can't just step back and not try and gloss over everything and say, oh, these are all safe, you know, because they might not be. Yes?
I'm not a scientist. I mean, I'm a cook, so I transform raw ingredients into a meal. And over the years, I've read things about, well, so-called junk genes. And it's like, well, they're junk genes, they don't matter. But then I read that, well, in fact, there's a lot going on with junk genes. They're not really junk. And then I read more about the instability of the genes. How does all of this play into the safety of the food? I mean, how does the instability affect, you know, from the
That is
genome to
such
the
a
plant?
great question. That is such a great question because basically what people, scientists have been doing is saying, all right, I've looked at this gene and how it's expressed in bacteria, and I publish papers on it. I know something about what's happening there. And when I take that gene out of bacteria and I stick that in a plant, it's going to behave just the way I anticipate based on its behavior here in bacteria, when we don't know. We don't know. We have this huge genome, and we're finding out things about the genome and how it's an active thing and it's changing, and there are regions of that junk DNA. Francis Crick named that junk DNA. And basically what he did was, well, only 3% or something of the genome encodes is a gene that encodes a protein, a protein, you know, encoding gene. Then the rest of that stuff is junk. And, you know, just because we don't know what it does doesn't mean it's junk. And would evolution, you know, as a biologist, I would think that, you know, evolution would be more efficient than that. That, you know, 97%, and we are finding, every day, they're finding more and more about that junk DNA, that there are enhancers that are at a distance from a protein coding gene that actually affect the expression of that gene, that if you change the distance between an enhancer and that protein coding gene, you can change the expression of that gene. So there's so much we don't know, and it could be affecting what we're doing with genetic engineering. And in fact, I heard a scientist once at Davis in the same breath say, oh, we know exactly what we're doing with genetic engineering, it's so precise, but really you should fund our research because then we'll figure out what the whole is happening with the genome. And I had to laugh out loud, but nobody else was laughing, and I just thought that's so incongruent, you know, the two different ideas. But we need to know more, you know, and the proponents will talk about being able to pick a safe spot that's not interfering with another gene, you know, a protein coding gene, for example. But we don't know enough about a genome at this point to pick a safe spot to, even if we could control where we inserted a gene. At this point, we haven't done enough research to know where a safe spot might be if we could put one there, a gene. So there's a lot of research that needs to be done. That's the bottom line.
So where are you working? Who hires somebody like you?
Well, that's a good question. When I left Calgene, I wrote my book. I had been saving newspaper articles and annual reports and whatnot, and I also was, I left when I was pregnant with my second child, so I thought, well, maybe somebody's telling me I should stay home for a while. So I was a freelance writer. I wrote articles for Orion and Nature Biotechnology and, you know, various science mags and other, Cooking Light, non-science mags. I did that for a while, and then I had established a reputation at Calgene of helping people write their papers. And so one of my colleagues, her husband is a researcher at UC Davis, and he had 25 papers he needed to get published, and so he hired me half-time to help him write and publish papers coming out of his lab, papers, theses, grants. And that was great because I could work at home and keep my hand in science, know what was going on, because he was a plant scientist. And then he ran out of money and couldn't afford an editor any longer. So I am currently working, I applied for a job in the Institute for Social Sciences at UC Davis. And actually that brings up another point. My husband kind of makes fun of me. He said, oh, science? Really? You're a scientist, not a social scientist. But I have come to appreciate social science. And in fact, I think one of the reasons why this debate over GMOs is so polarized is that we didn't incorporate social science at the beginning. We didn't bring in the economists, how expensive is this technology versus other possible solutions to the problem. How about human behavior? How are people going to respond to this? I think the sociologists and the economists and the political scientists, obviously politics got involved, but not, something we had any control over. I think it was a mistake for scientists to come out of the lab and say, trust us. We're the experts. We know this is safe. That was a mistake. It takes, when you, that's fine in the lab, when you got your project in the lab, but when it's going to land on somebody's plate, then you have to take into account other things in society. And society gets to weigh in on things other than the science. And so that's what I'm doing now. I'm working as an editor and helping social scientists write grants and get funded. But I look forward to getting back, maybe writing another book about. I can look back on the 20 years since the Flavor Saver, and where has this debate gone? Why? I think the social science of that would be interesting.
Vandana Shiva has an article in the September issue of Z Magazine. That's my level of scientific journals. She's not a fan of genetic modification, and she excoriates Bill Gates and the other billionaires that are backing Monsanto and Bayer and their technology. And one of the things they're doing to cover up making super weeds with their initial bad technology, and amaranth is one of the super weeds that they've created. And to now get rid of the amaranth, they're doing a gene drive technology that will eliminate amaranth from the genetically modified crops in the United States. But she says an unintended consequence of that is that it'll wipe out the amaranth that the global south depends on as one of their main nutritious foods. Do you care to comment upon that? How does that happen? Is this related to Terminator technology? Is this a realistic expectation?
It's not, well, I don't, is it related to Terminator technology? Terminator technology is a method for keeping farmers from reusing their seed. So in the United States, farmers don't usually reuse their seed because they're usually using hybrid seed anyway, and hybrid seed is not going to breed true. You'll get all kinds of variation in your crop if you try to plant your seeds the next year. The gene drive thing, that's pretty scary. I mean, one of the things, differences between genetic engineering and computer software is, plants reproduce themselves. They don't need our help. They get out there, they can handle it on their own. Whereas you got to have a hacker, or, you got to have a human involved in copying software. The gene drive thing, even the developers of gene drive say we got to look at this carefully, and we got to not rush out there to market with the technology. I don't know enough about the ins and outs of it, but I do know that it could be very scary if you made a mistake and you got it out there and it's reproducing on its own. The superweed thing, I hadn't heard that. I'll have to check out that article. I think different sources for articles are, I like to get my science from all different sources, so I think that's a perfectly fine source. The superweed thing that bothers me the most, and this, over the last 20 years, 20 years ago, you had your proponents saying, oh, the potential, we could do this potentially and that potentially, and we're going to feed the world, and we're going to do all these things. And 20 years later, they don't have a lot to show for that, besides superweeds and resistant, crops or bugs resistant to BT, which were the things that the environmentalists 20 years ago were saying, you're going to make superweeds. And Monsanto's saying, oh, no, no, no. And now the head of Monsanto is actually saying, well, yeah, of course we were going to make superweeds. We're selecting for it now. But to have made them and then turn around, the other thing they're doing, besides this amaranth you're talking about, which I'm sorry, I don't know more about it to comment more fully on it, is to, okay, well, we put glyphosate, made the plants glyphosate tolerant, and we made a glyphosate-resistant reeds. Well, now we'll just add another tolerance, make the plants, 2,4-D. Now we can spray them with 2,4-D, and we can spray them with glyphosate, and of course, the same thing's going to happen again, and you're going to have superweeds that are tolerant of both herbicides. You're repeating the same mistake. And I would like to think, this gets back to E.O. Wilson's quote where he studied ants, and he says, the thing about ants is they're not that smart individually, but they're really smart collectively. As a society, they're really smart. Humans, they're the opposite. They can be really smart individually, but collectively, we're not making very good decisions. And that's one of them. So we've made these terrible superweeds that are in 22 different states, and they're causing farmers all these problems, and they're in different countries, and it's a big mess. And the EPA steps back and says, and the biotech industry's solution is, let's do it again. And the EPA says, okay. That doesn't make any sense to me, as a scientist or even just as a logical thinking person. Yes?
I just was wondering, with your involvement and everything that you know about GE foods, do you eat any GE foods, and how much effort do you recommend the average person put into avoiding GE foods?
Okay, that's a good question. So I looked and looked for this talk. I took a photograph of my 2-year-old when the Flavor Saver tomato came out. And she's standing there holding their tomato. And I fed them to my daughter when they first came out. And I was pretty familiar with them. And, in looking back on it, I might not do that again. Because now Stephen Drucker had pointed out one of our other events that we commercialized, which I didn't talk about here. Early in the rat feeding study, a bunch of the rats died. And the company that we had hired out to do these tests for us, we didn't deal with rats at Calgene, just replaced them. And a couple of scientists who do those kinds of studies say that's not what you do. You do not just replace dead animals that die in the middle of your experiment, right? And the company said, well, it was gavage error. We're trying to jam a bunch of tomato down their gullets, and we killed them. So that one was, I think that got commercialized. That one that got commercialized. So in retrospect, maybe I wouldn't. But frankly, I try to eat organic as much as possible, and I feed organic to my family. But it's more kind of a protest. I think that the industry has gone in the wrong direction. They're not being above board like they should be, and it makes me angry. And so I'm not going to support them. And I think that organic, it's just more sustainable. Thank you. It supports, so far, most of the products. I mean, they wanted to save the world and feed the world, and they're not doing it. They have had 20 years and not much to show for it. And what they do have is supporting industrialized agriculture. And they have, they're selling chemicals that are probable carcinogens and spraying them all over the place. You know, glyphosate, when you, it's one thing when you are spraying it on the cracks in your sidewalk, but when you're spraying it over millions of acres of farmland all over the world, there are issues of scale that might come into play. And that has to be taken into account. So I try to eat organic as much as possible. They say 70% of processed foods contain some kind of genetically engineered ingredient. Yes?
As a, sort of a follow-up to that question, what is your current thinking about how best to feed the world today?
That's a really good question, because I've gotten to the point where I feel like I'm just being negative, you know? This is bad, this is bad. We got to go sustainable, and I would like to know more about agroecology, and instead of the old man-made techniques and man making nature subservient to man, you know, that we need to work with nature. We need to look at natural systems and soil bacteria and making healthy soils and all of that. So I would actually like to put my effort in a more positive direction like that. Because, you know, even the Green Revolution, Norman Borlaug and the Green Revolution guy said that, you know, it wasn't just supplying the seeds to the countries where the Green Revolution did have a positive effect, or at least increased productivity. You had to have the irrigation systems, you had to have the fertilizers, you had to have kind of the inputs that smallholder farmers around the world don't have access to most of the time. So we need to use more natural methods, but it's not easy. I mean, that's part of the problem. It's so nice to think, oh, if we just made the seed and we could just give it out to people in the developing world, wouldn't that be great? But it's not that easy. The devil is in the details, and we need to, it's more complicated, and we need to look at these other systems. And the UN says the same thing, you know, it's about sustainability. We have to not make dead zones in the Gulf of Mexico every year because of all the fertilizers that run off into the Mississippi and whatnot. So that's not a really good answer. We got to look into it. But I think the whole GMO movement, though, being so pro this technology that doesn't have all the answers, is preventing people from looking at the other possibilities, right? We should be looking at how to solve the problem. Would this method be best or that? And we're not doing that. We're not doing that at all. We're not even comparing how expensive the different possible solutions would be, and we need to do that.
And
Yes.
just on that note, really quickly, we've decided that we're going to continue this project in a more positive direction, like you said, and really bring in the experts on agroecology and focus
great.
on the solution. And because that's what got me into this, is just before I even knew anything about GMOs, was I was working with agroecological systems and, you know, advanced kind of inputs, quote unquote, not chemical fertilizers, but really interesting kind of inputs that really increased yield and nutrition and things like that. And then learning about GMOs and just how I already knew it was not working because I knew this other stuff was working so well and it wasn't getting the attention it deserved. So we're going to keep talking about GMOs next semester, I believe, and we're filling a roster of speakers right now that's going to take this discussion a lot more forward. So maybe you could be involved in that too, Belinda. Yeah. And did you have a...
I'd like to add, oh, never mind, that's fine. Sorry. Yes.
I'd just like to add. So I just recently retired from 35 years of organic vegetable farming here in the Northeast, right outside of Ithaca. I've been involved with Cornell around this issue ever since I moved here in 1987. I guess I would just like to interject that what we're needing to talk about is organic farming. And the conventional industry, conventional agriculture industry, Cornell included, has been subverting the term organics for the last 30 years, 35 years in getting us to talk about things like sustainable or natural systems or agroecology. And, you know, at every juncture, it becomes more confusing to the public. And, you know, as if we need to study it again. You know, organic farming is a fairly simple, intelligent procedure. And it's been being done for thousands of years. And it's a very, agriculture is a very manipulative process. You know, like a plowed field used to be a forest. And you just have to remember that. But if it's treated well, you know, it can produce food indefinitely. I mean, I ran my farm for 30 plus years on the same 12 acres and produced, you know, a lot of really great food year in and year out. And I didn't call it anything different since I started in 1981 until I just retired like a month ago. So
we know how to do it.
yeah.
That's awesome. Whatever we
Thank you.
call it, we know how to do it, and that's great. I remembered what I was going to say about your comment was part of the problem is because genetic engineering is so expensive, the grants have to be big to fund it, the research being done on the campus. And with the overhead numbers, you know, I think at UC Davis, the overhead is something like 57% of every grant goes to the administrators. I think that is a system that is screwing research into organic or adapting crops. UC Davis just got a big grant from the USDA to specifically breed crops for organic agriculture, because they're being bred for industrialized agriculture. And one of the reasons I think it is because they don't get the money. The organic breeders don't get the money because it's better for the university system to get the big grants that are going to do the genetic engineering stuff. So the system, we need to change the system in so many different ways, unfortunately.
Great, thank you for that comment. I just want to add too, that's something that I forgot to add, is we need to be very careful with agroecology and all these techniques, because if you type into Google agroecology light, L-I-T-E, that's kind of like a term, that's an article just came out kind of describing exactly what you were just describing, that this process is being subverted by a lot of the universities and the industries, because it's a buzzword now and people are trying to capitalize on that. And so people are, you know, talking about agroecology plus GMOs, and, you know, obviously that's incompatible. And you know, the industry really wants to get a hold of this buzzword. So yeah, we need to have a good, strong internal compass for this, where we're headed with this. And yeah, it's very simple. It can get very advanced, I feel, and incredibly nuanced and scientific with agriculture and, you know, all these amazing techniques that we have. Like a lot of people that know me know I'm really into this ocean mineral thing, which is what I got started on the farm I was working at years ago, where you pretty much dilute ocean water 100 to 1 with regular water. And I mean, it's a little more nuanced than that, but that's basically the process. And then you spray that on your crops, and you spray that on your soil, and there's studies that, you know, increases nutrient profiles dramatically in a lot of cases. It increases the beneficial relationships in the soil with the mycorrhizal and the bacteria. It can help you convert to organic farming within 1 year versus 3 or 5 years is often the case. And so that's like our most abundant resource that we have on the planet, and it could be, if not the solution, you know, a major part of the solution for moving forward. But that's still under the context of a very simple procedural way of farming that we've done for thousands of years. And thank you for your 35 years of farming too.
Thank you for your service.
Yeah. I have, if there's no more, I'll do the last one, I guess. It was kind of what James was talking about, a psychology question. I hope I'm not being redundant too, but you wrote a book on kind of the follies of genetic engineering and being somewhat critical of it with First Fruit. And this is something I ask myself a lot, not just with scientists, but with students and with people in society in general. What do you think, why did you write that book, and why aren't all scientists writing books like this? Why aren't more people standing up? Also, what does it take for a scientist to become an activist? Because I consider you an activist, and like Jonathan and Allison and everyone else involved in this project. Mm-hmm. And it seems like scientists today are very low on the activism kind of scale, you know? And
well,
so
they think it's activism to go out and promote the technology. That's
Okay,
their
so-
activism.
So okay, I see, yeah. And they think we're the terrorists, essentially. Yeah. But I don't know if you had anything to add to that. I mean, you already answered it really well with...
Well, actually, I wrote my book because we thought we were making history at Calgene. We knew we were going to be the first genetically engineered food out there, and early on in the process, I started collecting, memos and newspaper articles and whatnot, and I basically, I thought we were making history, and I wanted to record it. And it was something we said to each other, we're going to tell our grandchildren about this, and that's why I started writing the book. But over the course of living the book, my attitude changed. And while most of the book is, flat out, this is what happened, I also talk about the interactions among the scientists and try to make the scientists human, for the public and whatnot. They are human. They are human. And that's part of the problem, actually, right? The ants and whatnot. No, they have trouble giving up their dreams. But I added an epilogue to the book when things started getting, the movement started about questioning things more. And that I had people come up to me, former colleagues, who said, I loved your book, but why did you write that epilogue? That epilogue, what was that all about? And some of them only read the epilogue because the word got out that Belinda had written this book about the epilogue. Oh my God. So that's why I wrote the book, and my attitude had changed. But I think I'm a person that, I read something in the newspaper and I got to write a letter to the editor. Oh my gosh, they can't say that. So it's partly my personality that I see a wrong and I know something about it, and I can't stay silent. Staying silent is implicit agreement, is the way I interpret it, and I can't agree with the way things have been handled, and so I have to speak up. And the bottom line is it's a conflict of interest for most scientists, and frankly, I'm free because I'm a writer and an editor now, and I'm not doing science. That my livelihood is not necessarily on the line. They could be mean to me, but so far so good.
Yeah, it seems you really have to step out of the system to really talk about it at all. I think
so.
And now, what percentage, just really quickly, of scientists, if you had to ballpark it, would you say, no, there are problems with genetic engineering, but remain silent, versus is it just all scientists are in the dark about this, And what would you say as being coming from the front lines?
Most genetic engineers I know, academics, know these issues. Most of them know. And they either stay silent because they don't want to rock the boat, or they're promoting the technology because it's their bread and butter. And they believe, they're still holding on to their dream. So it's a large percentage. There are very few people who come out of the shadows and say, I really like that letter to the editor you wrote, or, the New York Times letter to the editor, or, the letter to the New Yorker, or whatever. I get a few, but mostly they're staying silent. And it's a lonely place to be here, actually. So I'm so glad to be here among some like-minded people.
Thank you.
You're welcome.
Yeah. All right. Thank you, Belinda. Yeah. That was amazing.